All right, so let's start. This chapter one is all about cells. All right. And in the IMAT syllabus, you have to know the anatomy of cells. So basically, what's present inside the cell, what are the different types of cell, and the general structure of our cells. So in chapter one, there are two types of cells that you need to know, the eukaryotes and the prokaryotes. Now, eukaryotic cells are found in humans, fungus, and animal cells. We humans have animal cells, and these cells are all eukaryotic cells, whereas prokaryote cells are generally cells that are found in bacteria. So these are the differences that you need to know. And second thing is that our cells, the eukaryotic cells, are multicellular. What do I mean by multicellular? Well, us humans are made up of a lot of different types of cells. So we're not made of one cell. This one cell is not human. A lot of these cells, millions of these cells make up us humans. So we humans and all eukaryotic cells are multicellular, whereas unicellular is when you have a single cell. When I have a single cell, I call it a prokaryote. So if I found a bacteria, this bacteria would be made up of only one cell, not a lot of cells. So we call this unicellular. All right. And the third difference is that eukaryotic cells have membrane-bound organelles. What do I mean by organelles? Organelles are the organs of our cell. So just like us, humans have different types of organs. We have kidney, we have stomach, we have kidneys. Just like that, our cells also have organs, and we call these organs organelles. And inside, and these organelles are surrounded by a membrane. They are membrane-bound organelles. For example, let's say this is a nucleus. This is surrounded by a membrane, and I'll talk about that later. But for now, you need to know that eukaryotes are membrane-bound organelles. Prokaryotes, they do not have organelles that have a membrane. So it's not membrane-bound. And prokaryotes also do not have any nucleus. Eukaryotic cells, we have a nucleus. That's another difference you need to know. And finally, prokaryotes, all of prokaryotes have DNA, ribosomes, and cytoplasm. No matter what type of cell the prokaryote is, all of the prokaryotic cells will contain these three things. And this is a very popular question they can ask you. So they have a DNA, they have ribosomes, and they have cytoplasm. All of this I'll explain later on. So these are the few differences that you need to know. And now we move on to the different types of organelles. So what you see here, all of these are something what we call organelles. And this is an animal cell, all right? Now, the next thing which is outlined in this syllabus is that you must know the different types of organelles and their structures. So I will start with the most famous organelle, which is the nucleus. Now, nucleus has DNA and mRNA. It is the place where our genetic material is stored. And this DNA, right? This is a DNA. Don't mind my drawing, but this is a DNA, and this DNA is wrapped around histone protein. So I've wrapped this DNA around histone protein, and what this does is it reduces space. So the DNA is wrapped around histone protein. So DNA would combine with histone protein to reduce space. All right. And when these two combine, I call this a nucleosome. In simple terms, DNA plus histone gives you a nucleosome. And I'll talk about nucleosome later on, but for now, you need to know what a nucleosome is. And this nucleus is surrounded by this membrane. We call this membrane the nuclear envelope, all right? And this nuclear envelope has Pores. Pores are these small holes through which DNA and mRNA can escape. Now, that was all about nucleus. There is this dark spot inside the nucleus called the nucleolus, all right? And the nucleolus has two main roles. It contains dense pack of DNA and proteins. So the nucleosome, the reason that it's darker than the nucleus is because it has a lot of DNA and protein. And it has another function, which is it makes ribosomes. Ribosomes are proteins that make other proteins. Guys, if you can close your camera. So this was the nucleolus. So when the nucleolus makes ribosomes and DNA, the DNA escapes through the nuclear pores. All right. Now comes mitochondria. This right here is mitochondria. And you need to know that the mitochondria is the powerhouse of the cell. What this means is that it makes energy. So the role of mitochondria is it makes energy in the form of a molecule called ATP. And you learn more about ATP in second year A level, which will be on, I think, Tuesday. But other than that, it is double membraned. Remember this, that in all of the organelles, only the mitochondria is double membraned. Apart from that, all of these are single membrane. So this is a double membrane organelle, and its role is that it makes energy for the entire cell. Now comes the rough endoplasmic reticulum, or what I call RER. The reason that it's rough is because it contains ribosomes. So these small dots that you see on the rough endoplasmic reticulum is ribosomes. So the role of the rough endoplasmic reticulum is that it folds and packs proteins made on the ribosomes. So the ribosomes that are produced, the proteins that are produced on the ribosomes will be packed and folded on the rough endoplasmic reticulum. That was the role of rough endoplasmic reticulum. And right next to the RER, you will find another organelle, which is very similar to this rough endoplasmic reticulum, and it's called the smooth endoplasmic reticulum, or SER. So the SER has no ribosomes. That's why it's smooth. And its role is that it will pack and process lipids, steroids, and hormones. That's all you need to know about SER. Then comes a very important organelle called the Golgi body, or some places you will find it called Golgi apparatus. So the Golgi apparatus, which is this right here, this is called the Golgi apparatus, will pack and process proteins, lipids, and also make lysosomes. So it will pack the proteins and lipids and then send it out in these Golgi vesicles. And it makes another molecule. It makes another molecule called the lysosome. Now, lysosome are also these vesicles which contain uh, enzymes that break down unwanted organelles. So let's say, for example, one of the organelles are no longer needed, then the lysosome's job is to break down unwanted organelles. So it will engulf the organelles and destroy it. Now, centriole is another important organelle, and you'll find the centriole right here. So these two are called the centrioles. And the centriole's job is to, it's involved in cell division. That's for now, just know that it's involved in cell division. In A2 biology, you learn more about centrioles, but the two things that you need to know is that it is involved in cell division and it contains something called microtubules. These are the microtubules, and later in the life class, I'll tell you more about centrioles. Then comes the centrosome. Now, centrosome are also centrioles, but centrosomes are two of these centrioles combined together at right angles. So when two centrioles, they come together at right angles, they make centrosome. And that were all the organelles that you need to know for AS biology. Now, there are two important organelles that I want to talk about, the centriole and the ribosomes. So these dots that you see in the cytoplasm are called the ribosomes. Now, let's start with the ribosomes first. All right. So what are ribosomes? Ribosomes are proteins that convert mRNA or genetic material into proteins. So, for example, DNA in the nucleus will be converted to mRNA. You don't need to know more details about this, but you need to know what this is called. This is called transcription. So the process where DNA is converted to mRNA is called transcription. And when mRNA, this part where mRNA becomes protein is called translation. So when mRNA becomes protein, this is called translation. And this process happens at the ribosome. So mRNA being converted to protein is called translation, and it happens at the ribosomes. So the job of ribosomes is to make proteins. Now, where are ribosomes found? It is found in chloroplast in the plant cell, mitochondria in the animal cell, and cytoplasm in both animal and plant cells. Now, there are two types of ribosomes, the 80S and the 70S. Now, these 70S ribosomes are found in chloroplast and mitochondria, whereas the 80S ribosomes are found in the cytoplasm. So let me just go back here. So all these ribosomes that you see here, they are the 80S ribosomes. So the ribosomes that are found in the cytoplasm are called the 80S ribosomes. The 70S ribosomes are found in the chloroplast and mitochondria. Now, you need to know the structure of the 80S ribosome. So this right here is an 80S ribosome. And 80S ribosomes have two jobs. 80S ribosomes have two structures, the 60S and the 40S. So the 60S is the bigger part, and the 40S is the smaller part or the smaller unit of the ribosome. And they come together in protein synthesis. So these two, they come together in protein synthesis. All right. And that was pretty much what you needed to know about ribosomes. Now, I told you the second organelle, which is the centriole, is also very important. And this is a very popular question, the microtubules, which will come. And you need to know how the centrioles are made up of. So the centrioles, right, are made up of microtubules. Now, what are microtubules? These rods that you see here are called microtubules. And their function is that they give the cell shape. So microtubules act as the cytoskeleton. Uh, I don't know if the spelling are right or not, but microtubules give the cell shape and acts as a cytoskeleton, right? And it also makes the centrioles. Now, what are microtubules made up of? They are made up of a protein called the tubulin. And there are two types of tubulin protein, the tubulin A and tubulin B, or in simple words, the tubulin A is called tubulin alpha and B is tubulin beta. And when two of these tubulin, so tubulin alpha and beta, when they combine together, they make something called the dimer. So what is a dimer? A dimer is simply one alpha tubulin and one beta tubulin coming together. This makes up a dimer. So 13 of these dimers, when this is one dimer, right? So when 13 of these, they combine together. So this is one dimer, this is another dimer, this is the third dimer. So when all of these, they come together, they make what we call one microtubule. So basically, let's say this is a microtubule. It is made up of tubulin, right? And tubulin is made up of dimers. So this is one dimer. And when 13 of these dimers, they combine together to make this microtubule. Now comes the very important part. This thing right here. So 9 groups of microtubules make up the centriole. What do I mean by groups? So one group consists of 3 microtubules, right? Let's say this is one microtubule, this is the second, and this is the third. So 3 of these, they combine together and they make one group. So when 9 of these groups, they come together, let's say this is one group. Similarly, 9 other groups will come together and they'll make one centriole. This is also written right here. So this part right here, where centriole, each composed of 9 microtubule triplets. So one group is called a triplet. And 9 of these triplets, they come together to make centrioles. Let me explain this once again, because this one is a very important concept. Centriole is made up of microtubule. Microtubules give cell shape and acts as a cytoskeleton. Microtubules are made up of tubulin protein. There are 2 types of tubulin protein, tubulin alpha and tubulin beta. And when 2 of these combine together, when one alpha tubulin and one beta tubulin, they come together, they make a dimer. And 13 of these dimers make up one microtubule. And when 3 of these microtubules, they form something called a triplet. And 9 triplets, 9 of these triplet groups, they make one centriole. This was centrioles, and please remember this again and again, because this is a very important concept. So the next thing is plant cell. The cell that you learned earlier, this cell right here, was animal cell. And now comes the plant cell. So for the IMAT syllabus, you need to know the difference between plant cell and animal cell. Basically, they'll ask you what organelles are present in the plant cell and what are present in the animal cell, and what are not present in the animal cell compared to the plant cell. So these organelles that you see here are present only in plant cell. So these organelles are only found in the plant cell and not in the animal cell. What are these organelles? Let's start by the vacuole. So you've already heard about the mitochondria, the cytoplasm, and then what else? The ribosome, the nucleus. These are all the organelles that were found in the animal cell as well as the plant cell. So I'll not talk about these, but what I'll talk about are these organelles right here. So vacuole, this large vacuole is an air sac, and it contains sugars and proteins and some ribosomes as well. Its job is to act as a storage. So it will store all the sugars, the proteins, sucrose, ribosomes. This will be the place for storage. And it has another function as well. This large vacuole will push these green organelle, which are the chloroplast. So large vacuole will push the chloroplast to the end. And what are the chloroplast? These chloroplast will trap light energy. So light energy will be transferred, and light energy will be used to make ATP. So this ATP is made by the chloroplast in the plant cell. And in the animal cell, which organelle makes the ATP or the energy? It's the mitochondria. So in animal cell, mitochondria makes the energy, and in the plant cell, the chloroplast makes the energy, both in the form of ATP. So we're done with chloroplast, vacuole. Now comes the cell wall. Now, the cell wall is found below the cell membrane. So it's outside the cell membrane, and its job is to provide strength and support to the plant cell. That's one of the functions. The other function is that it protects the plant cell from foreign substances. So not everything can go inside this plant cell thanks to the cell wall. That was the cell wall. And finally, this organelle called the plasmodesmata is found only in the plant cell. It's not present right here, but it has one main function, which is that these plasmodesmata acts as bridges between organelles. So let's say I want to transfer ribosomes to the mitochondria. This organelle will help in doing so. So this acts as a bridge between two organelles to transfer substances. That's what plasmodesmata is. That's everything you need to know. I don't think there is anything else for chapter 1. So the difference in plant cell and animal cell, the anatomy of plant cell, and the anatomy of animal cell as well, and the anatomy of animal cell as well. And then you also need to know what ribosomes are and their function, what centriole are and what microtubules are, and finally, the anatomy of plant cell. So let's do this exam question. This is from IMAT, all right? So I'll give you around 2-3 minutes to answer in the chat. And if you can't, then I'll explain it myself. You can always answer in the chat. Yes, uh, it is ribosomes, everyone. Good work. So, what are always present in prokaryotic cells and not eukaryotic cells? Capsules are only found in some bacteria, and flagella is the this thing right here which helps cells movement. So when the cell wants to move, this flagella will help with that. And also, it's not found in all prokaryotes. It's only found in some bacteria as well. Pili, you don't need to know what it is. It's not found in most of the bacteria or the prokaryote cells. Now, ribosomes are found in all prokaryotic cells. Why? Because ribosomes make up protein, and proteins are the most important thing in all of the cells. So proteins make up all the cells. It will help with the cell function, with everything. So ribosomes make proteins, and proteins are found in all prokaryotic cells. And I've also mentioned it, I think, right here in the starting. Yes, right here. So prokaryotes, they contain, all of the prokaryotes contain DNA, ribosomes, and cytoplasm. And DNA is the genetic material which is found in all cells. And this is very important as well. Ribosomes, I said it earlier, very important. Cytoplasm is the liquid in which organelles flow in. And this is also very important. So it's found in all prokaryotes and eukaryotes as well. And I think that was all what you need to know about chapter 1. So in the next class, I'll do chapter 2 of AS Biology, which is biological molecules. In that, you will